Saccharomyces cerevisiae, or baker's yeast, has been the most widely used and oldest microorganism used in biotech. Now, in ancient times, baker's yeast has been used to ferment bread, beer, and wine. But now, in modern times, it's actually been used in large-scale industries where they produce certain types of chemicals, pharmaceuticals, and biofuels. In the case of the pharmaceutical industries, baker's yeast have been used to produce large amounts of insulin, human serum albumin, and certain types of vaccines, such as those against HPV, human papillomavirus, and HIV. Here at the University of Philippines, Diliman, at the Institute of Biology, to interview certain students and professors as to regarding what they know about yeast as the model cell factory. You know what a microbial cell factory is?
A microbial cell factory is an approach to bio-based engineering wherein live microbial cells are used as a production facility. Microbial cell factories convert a basic or intermediate compound into the desired chemical for industrial. The biggest challenge of using the microbial cell factory is that the conversion process must be fast, efficient, and cheap. Thus, may require optimization such culture media optimization for the production of the native compound or gene overexpression to increase product yield. In addition, genetic and metabolic engineering in a directed fashion can be done in order to create new properties and new products in doing the conversion process.
What are yeasts and what makes them special for the industry?
Well, yeast are preferred to be used or are attractive for use in the industry since they are very well characterized and its genome is available in different online databases. For products needing post-translational modification for its bioactivity, yeast cells can carry out numerous post-translational modifications such as phosphorylation, O-link glycosylation, N-link glycosylation, acetylation, and acylation. It is also genetically easy to manipulate. Moreover, it is generally regarded as safe, which means that products produced by yeast is easy to put out in the market. Lastly, it is a robust industrial organism and it is thus easy to scale up and be used in the industrial setting.
New products produced by yeast.
One product that I know of that is not native to Saccharomyces cerevisiae is insulin. In the early 1920s, diabetic patients were treated with insulin purified from bovine and porcine pancreas. With the development in the field of genetic engineering, recombinant human insulin can now be produced in a yeast-based expression system which produces soluble insulin precursors which are secreted into the culture media supernatant, making it easier to harvest. For the expression of the recombinant proinsulin in yeast, insulin construct was engineered to contain the native A chain and B chain lacking the C-terminal B30 threonine. The cDNA sequence encoding for this construct was fused with an alpha factor signal sequence from S. cerevisiae from secreted expression of proinsulin. This process gave yield up to 80 mg per ml, which is industrially significant. The resulting proinsulin is then converted to active insulin. Aside from this native recombinant insulin, other analogs are also being produced by yeast such as insulin aspart and insulin detemer. I think that's all I know about insulin from yeast.
Examples of what yeasts can produce.
S. cerevisiae is one of the widely used and studied microorganisms for the production of ethanol. It has been employed in the industry by converting cheap sugar-rich substrates such as molasses to ethanol. Native high-producing strains are industrially used. However, several methods have been used to improve ethanol yield with the use of conventional methods such as protoplast fusion and genome shuffling, and directed evolution methods such as the use of error-prone PCR to engineer yeast RNAP subunit to improve yeast ethanol tolerance and production. Yeast pathways have also been engineered to produce isobutanol, a technology which is already implemented by Gevo Incorporated. Studies with yeast as cell factories for the production of carboxylic acids and derivatives, terpenoids, and other industrially imported compounds are already underway.
So, any products produced by yeast?
Are you familiar with artemisinin? So, artemisinin is an antimalarial drug precursor that is produced by the plant Artemisia annua. Artemisinin derivatives, although designed as first-line anti-malaria drugs by WHO in 2002, was hard to come by because of its limited availability, which was due to the months of lag between initial requisition and eventual supply, which was dependent on the weather and overall harvest of Artemisia annua. As a result, both supply and price of the drugs have fluctuated substantially over the past years. Thus, the semi-synthetic artemisinin project was initiated with the aim to provide an alternative source of artemisinin to stabilize supply and price of the artemisinin-based therapies. And by 2013, they were able to complete the artemisininic acid biosynthetic pathway in Saccharomyces cerevisiae by the use of the yeast native mevalonate pathway and the expression of the key genes from A. annua, such as the artemisinic alcohol dehydrogenase or ADH1 and artemisinic aldehyde dehydrogenase or ALDH1. They were able to optimize and balance the enzymes to increase the amount of artemisininic acid. This new yeast strain was also engineered to utilize glucose such that the precursor of artemisinin, artemisinic acid, can be produced by the yeast from glucose.